thanks very much for giving me this opportunity to tell a little bit about our program. It spans quite a few years. We have been, for a long time, I've been collecting genomic resources, so we're only finally, in the last little while, starting to put things together, and I think it's certainly for the North American leaf rust population, it's becoming quite an interesting story. So, as the title suggests, I'm focusing on leaf rust, but some of the things I've also been can be applied to the other rust, stem rust and stripe rust, and I have been involved in some stripe rust projects with my collaborator Gert-Jan Brar and his bioinformatician Sam Holden. Leaf rust is the most prevalent worldwide, although it's not always as obvious. If we look at some data, this might already be a little bit outdated from Savory et al. Their leaf rust was globally put at the most, causing the most losses. But if you see here in North America and Canada, it's a little bit less because it's quite well contained because of use of genetic resistance and fungicide use. But globally, it's causing a lot of problems. Stripe rust is down here, stem rust is here, and other diseases. So as I said, it's quite well controlled in North America and also in some other places, but here, because we have good genetic resistance built in, and of course there's a lot of fungicide used mainly to control Fusarium head blight, which also controls then the rust. So sometimes it's a little bit tricky to estimate exactly how well the resistance in our wheat cultivar works for the leaf rust disease. Many genes have been discovered, and just to put in a little plug here for the late Peter Dick, who discovered a lot of the genes for leaf rust. So as you will know, most leaf rust resistant genes are race specific and therefore are only effective against a single pathogen isolate. Here's an example of an isolate that causes pustules, so it is lacking a resistance gene. In this case, this isolate on a Thatcher genetic background which has the LR2C gene is causing necrotic lesions. I'll just show a little bit of this background because not everybody works on rust diseases. There's a number of leaf rust resistance genes that have been cloned, mentioned here, and the majority code for the sort of classical NBS LRR genes that are involved in direct recognition of pathogen vectors in the cytoplasm, most likely. So for that, it is important that we actually also focus and try to clone these effectors to help understanding the interaction and also to assess the effectiveness of these isolates, and if they change in the populations, what is their prevalence and how often do they change? What's the risk? So we would like to know much more about these effectors and how they are distributed over the populations, and also the mechanism of action. There is also non-specific, more durable resistance genes in LR34, 46, and 67. It's multi-pest. It's also controlling other rusts and powdery mildew, even FHB, Fusarium head blight. It's called also slow rusting genes, and often they're used in combination with the single race resistance genes.
I will ask him.
Some of them are cloned, the LR34 and LR67. It's already a while back. And then when they used in combination with the single race-specific resistance gene, they really cause, or they are really quite durable. So this is more the research of my collaborators, Brand McCallum, Colin Hebert, and Kurt McCartney. So I'm not going to talk about that. Maybe they can talk another time in this series to explain their work and how that correlates with what we are doing. So more to the topic then of this talk I'm giving is the impact of genomics on the rust research. It has been quite profound. As you know, the rusts are biotrophic pathogens, and you cannot grow them on plates or in culture. So when we started sequencing and getting information on gene contents, that was really a game changer. So originally, molecular markers for these rust isolates were developed. A lot of this work was done by Jim Colmer and others, also in Canada by Brand McCallum and Shiben Wang. And I was involved in that in the beginning. This was based on SSRs, rapids, and AFLPs. But soon after, when I started at AFC in 2000, we got money through genomics projects and programs. And we started working on sequencing. In the beginning, it was ESTs through Sanger sequencing, which also allowed proteomics research, work by Chris Rampich and others. And then a little bit later, the next generation sequencing, Illumina sequencing, allowed us to get transcriptomes. And the sequencing of genomic DNA then allowed us to get many more markers per isolate. So allowing a much better resolution of the phylogeny on these isolates. And I'll talk about that in a minute. Of course, the first genome sequences for rust is the stem rust and poplar rust, published in PNAS in 2011, and by us for race 1 of the leaf rust, Puccinia triticina, in 2017. Those were important but interesting, and I should mention that here they were what we call collapsed genomes. Since the basidiomycetes have 2 nuclei, or dikaryotic, and the genomes of these 2 nuclei appear to be quite variable. Hundreds of thousands of SNPs are recognized between these 2 haplotype genomes, and also we see lots of structural variations and transposable element changes. So when we got this DNA and sequenced that, the software at the time couldn't really distinguish very well between some of these regions that were too similar in the 2 haplotypes. So we got collapsed genomes. Of course, the sequencing of wheat genome was important as the host. I don't really want to talk much about that. And then more recently, long read sequences, Nanopore, PacBio, HiFi, and HiC really allowed now the haplophasing, so resolved dikaryotic nucleic genomes, starting in 2018 with several groups in Australia, Ben Schlesinger and Jonas Perschneider, Peter Dodson, Melania Figueroa, and also in China on stripe rust. And also important was the development of pipelines now for identifying effector candidates. And since then, several evirance effectors have been cloned, some of them by mutation analysis. So just a quick reminder of the life cycle. Maybe not everybody is familiar with that. What I should point out here and what I mentioned here is that in the most of the life cycle, the fungus is dikaryotic, and this holds true for many basidiomycetes, if not all. So 2 separate nuclei, which are maintained throughout the life cycle, except for when pterospores are formed, and upon the germination of those, goes through meiosis and forms those haploid progeny, which infect the alternate host. Fertilization of the 2 mating types, of the 2 different spores. I should mention that the mating type is also segregating now in this during meiosis. I'll come back to that in a minute. But if there's compatible mating types, fusion will happen and the dikaryotic state is being reestablished, and that's these production of spores on the alternate host can then reinfect the main host, at least in agriculture, the main host, which is wheat or grasses. So the life cycle on the primary host is relatively short for leaf rust. Within 7 days, you can often start seeing pustule developments. We have extensive transcriptomes of these stages on 3 different interactions, but I'm not really going to talk about that today. In North America and in other places in the world too, there is patterns of spread of the fungus, and in North America there have been 3 main currents identified. The main Paccinia pathway coming from Mexico through the Midwest into Canada here, and on the west coast, on the west coast, the west side of the Rocky Mountains, there is a different current, and I'll tell you in a minute why that is interesting. And there is also another current which might be more spreading disease propagules, in this case rust spores, along the eastern seaboard into Canada. So as I mentioned, several groups, including Jim Colmer and Brent McCallum and Xibin Wang before, and also other people in other places in the world, in Australia and South Africa, have looked at how these isolates relate to each other using phylogenetic analysis, initially with using SSRs and AFLPs, as I mentioned. But lately, we have now with the next generation sequencing, we have now many more markers, and it has been easier to get higher quality phylogenetic analysis done. So here we're building on initial paper several years ago we did with John Fellers and Jim Colmer. But now we have looked more carefully into a Canadian population from over the last 5 years from all the different regions in Canada. We also included some Mexican isolates now, and we took some public available data, as I mentioned, from our study, John Fellers and Jim Colmer, but also from several Australian isolates. And we did both genomic DNA sequencing and RNA sequencing. The RNA sequencing was basically based on the work that has been pioneered by Diane Saunders on pathogenomics, so basically taking infected wheat leaf pieces and sequencing RNA from that. And we did that for several reasons. First of all, we could get a better idea of the expressed effectors during the host infection. And when we map, we can indeed get many gene covers, up to 10,000 genes from these samples, just from sequencing one sample, 20 million reads for $150 US. You get a lot of information from that. We also did that because it was, as a cheaper and quicker. You don't need to purify your isolates. And what's interesting how here we have a phylogeny tree, and on the outside you see the red and the blue, where red is sequences derived from RNA sequencing and the blue from DNA sequencing. So you do see some clustering, but we did another filtering and analysis of why that might be. One of the things which is because we have haplophased genomes of representatives of the different clades. My bioinformatics student, a very talented guy, Sean Formby, was able to haplophase the reads. And what we could see is that when you do RNA sequencing, see that not all genes are expressed from the same haplotype. So for that, we have to be careful when you do phylogenetic analysis, where you mix SNPs called from RNA sequences and DNA sequences. So, but we got a handle on that, and we are pretty confident that you can mix the isolates or the markers derived from the different sources if you do the proper filtering. So what you can see here, just in general, is several clades, about 4 or 5 major clades in North America. There's 2 major clades and then some smaller ones here. But we also found one here, for instance, which was basically quite specific to the Pacific Northwest and British Columbia. So that might be indicative of different populations existing in North America and in Canada. So it is still useful to sample and look at the whole population in different regions, and not assume that because of the introduction or the major flows of the current wind currents, you get the same populations. There's another example here where when we sequence more, originally this was thought of one population, it's splitting out. Now what's new in this analysis is that we also looked at the mating type genes. And I should just explain that a little bit. I've been interested in mating types for a long time. It started in when I was a research associate in Jim Cronstedt's lab, working on the mating types in Eustelago, comparing Eustelago metis and Eustelago hordiae. And the paradigm in Eustelago metis is or the mating type genes is the best studied examples. What was found at the time is that apart from the pheromone receptor and pheromone mating type pheromone genes, you also have this 2-component system, which are homeodomain containing transcription factors, which we called in Eustelago metis B West and B East, and they're divergently transcribed. This whole unit is about 6 kb. So there is an intergenic region here, which should contain both promoters for East and West. So it's within a kb. But what was found at the time that there is different alleles in different isolates of Eustelago metis, whereas in Eustelago hordiae, we only found 2 alleles in nature, as far as we could tell. And we figured out that this was because in Eustelago hordiae, the mating type genes are on the same chromosome, so contained on a so-called sex chromosome. But in Eustelago metis, they're on different chromosomes, and that sort of allows the mating type genes to diversify and become multi-allelic. And that's thought of as during evolution, it increases the chance that during the sexual cycle and when they have to find a mate, that the chance of getting a compatible mate is increased. So because normally, as I mentioned, the basidiomycetes are dikaryotic, it's thought that the B West allele of one mating type interacts or forms a heterodimer with the allele of the yeast allele of another mating type specificity. And that then forms a transcription factor that starts a whole program of genes that are involved in filamentous growth in Eusthalagomatoes. Now, in rust, we don't know for sure what these are doing. We assume they have the same function. And in 2017, we did check their function by using the Higgs approach. So this is the host-induced gene silencing approach, where we silence one of the mating type genes, and we did get less virulence during infection. So they probably have an effect during the growth of the fungus. Nobody has yet looked whether they're involved in mating. That's most likely. But anyways, this is just to let you know that in rusts, we also find them on 2 different chromosomes. So it's a tetrapolar organization. And therefore, there is also different mating type specificities found. Sorry. So when we look at the, when we look at the phylogenetic trees here from the molecular phylogenies of the B alleles for leaf rust, stem rust, tripe rust, and oat rust, they all fall in the same clade, so all the B west and B east, which indicates that there's a very ancient predecessor that had these different modules, and that within the species they have been diversifying. So now back to this phylogenetic tree then. So we did analysis of all these isolates, and we do find 6 alleles, so different specificities within the North American population, but worldwide we're now up to 11. 5 had already been found by Jana Spersnyder a couple of years ago, and Ben Schlesinger also added a few. We're now up to 11, but 6 are represented in only 6 in the North American population. But what's interesting is that if you look at the combination of the mating types, it follows the clades. Now, this might not be surprising. It's been known that leaf rust is mainly clonal. The sexual host or the alternative host is not available in North America for leaf rust, and in several other places neither. So it's mainly thought to be present in the Fertile Crescent, Turkey, Israel, Syria, and around the Mediterranean basin. So this would confirm that it's really the mating types are contained within these clades, but not everywhere. You can see that there is different combinations, which is really interesting because that indicates that there is different introductions or possibilities, that there were introductions and they formed or been evolving as clonal lineages within certain regions. We also looked at some here of the durum isolates, Ethiopian durums, which are considered in the origin of diversification. European durums are more evolved from that and derived from that, and this is between parentheses because this also includes durum isolates that are found in South America and Mexico. So they've probably spread, and that's something that Jim Colmer has already touched on earlier in earlier publications. Interesting though, we see quite a variety of mating type genes here. Also in some of the European, Turkey, Israel isolates, there's more diversification. So 2 years ago, we did the same in stripe rust populations. That was in collaboration with Sam Holden and Kucharn Brar. And they also found more variation of the mating type alleles in isolates from China, the Himalayan area, so which is considered the region where diversification of stripe rust takes place. So now having this kind of information is really interesting to see what we can do with that. So to look further into population structure, Sean Formby did an excellent job in phasing the SNPs that we found and looking at more particularly the population structure using admixture now with pruned SNPs. And we can indeed see now that we see this nice bifurcation of the population structure, almost like you can already predict of which haplotype you're having. In this case, you see the mating type 4 is very prevalent in North America. This is this salmon-colored haplotype SNP collection. And it is here linked with MAT1, which is this blue. But in this plate here, it's not the same blue. So this is already an indication that this mating type 1 is probably derived from a different isolate at the time. And we have a few other examples where it's not holding up exactly. So what that means is that the original strains probably derived through meiosis, genetic recombination, and maybe were reintroduced or introduced from different isolates, and then may have recombined through somatic exchange. So this was discovered a few years ago by Peter Dodds and Melania Figueroa in stem rust, where the nuclei can be exchanged through somatic hybridization and cause new virulent phenotypes, because the different haplotypes might have different combinations of virulence and avirulence genes. So at this point, we're still figuring out exactly what's going on and how we'll go from here, but this kind of analysis, because we have a lot of gene information, will really allow us to look more specifically at through genome-wide association studies with virulence profiles to see what's going on and to try to identify specific alleles of the effectors. So we have now also haplotype, so phased haplotype genomes from isolates representing all these different clades. And we're trying to sort this out in the next half year. One of the things that came out, or that we started with, is when we found out that we get these nice identifications of mating type alleles, whether we can use the MAT alleles as proxies for haplotype. And the answer is it can be for certain clades, but there is exceptions. So we need to find other markers to get a quick handle on what certain lineage we have if we have found a different isolate or analyzed a different isolate. Nevertheless, we did start on a project here. This is work from Janess Holmes, where we have a multiplex qPCR assay now for all the 6 isolates, 6 mating type alleles in the North American population using qPCR and fluorescent probes. And we can distinguish all 6 in a very quick, easy assay. And the same we're doing for stripe rust. But as I mentioned, now we need additional markers to pinpoint different genetic lineages. But since we have this information, we can probably design some other markers so that in a relatively quick multiplex assay, maybe a couple runs, we can, if we get an unknown sample, we can identify from which lineage it is. We also have isolates from the different time points. So we're trying to see if we have enough information that can sort of look at the evolution of the different mating type and different nuclear combinations that we see in North America. But we might have to sequence more of older isolates because, as I mentioned, we have a good collection for Canada for the last 5 years, but not from some of the other regions. And a lot of these isolates, we normally get information from our DNA sequences from John Fellers or Jim Calmer. So the idea then is when we have some interesting and reliable assays that we could use that maybe in a kind of a biovigilance approach. So over the last 6 years, we did a study which was just published recently where we had a few spore samplers in different places in British Columbia and Alberta. So that is the southwest corner of Canada. And these are Burkhardt samplers, so continuous vacuum samplers that suck up whatever is in the air. And we were at that time just looking at the airborne microbiota and see if we can identify those and see if we can find rust species in there. Here we only used the ITS2 sequence, which was PCR amplified and Illumina sequenced. This was work that was done by Wen Chen and Sarah Hamilton, also in collaboration with Andre LaRoche in Alberta. They have specific pipelines that could identify very quickly the ITS sequences and assign them to different fungal species. I pointed out here Puccinialis, which are the brown here, and this is the different sample sites that we used. And then more specifically looking at which Puccinialis we have, the genus Puccinia here is in purple. We could identify that. Then more specifically, it was identified with specific PCRs to see whether we had the correct species, because some of these leaf rust and stripe rust and stem rust are present in different clades that are not always different or pathogens on different grass hosts and different are present in different areas. So you have to be very specific with your analyses. But nevertheless, we could also look then over time at the different locations and see the dynamics over the season. And finally, we also looked at the patterns and trajectories using modeling of air currents. And this was done by Nathaniel Newlands in our group. And we did find indeed that some of the peaks that we find in areas could be explained by certain currents and model or trajectories of wind packages, if you will, over the time period leading up to certain increases that we found in our spore samplers. So this is just to illustrate that you can do these things. This was just a pilot experiment, but with the assays we are developing, we're hoping to pinpoint more precisely in spore traps whether we have certain lineages of in this case, leaf rust, and can therefore maybe have more predictive value of what we're seeing in our wheat growing areas. So I will talk a little bit now about exploiting then effectors, what we can do with them. So when you find effectors, they are probably interacting with resistance genes directly, at least for some of the examples we have from the rust AVR genes and R genes. So having fungal effectors will help with to be able to clone the specific R genes, and also maybe assist us in the classical breeding by screening progeny. We should be paying more attention, and once we have AVR genes, we can screen the populations for genotypes and determine the risk of avirulence to virulence. Often it are likely the AVR genes, if they are in a heterozygous state, they can mutate easier to a virulent state. So if we have that information in isolates, we can assess the risk better, and also whether certain genes that are deployed in certain cultivars and grown in certain areas are more at risk for introduction of certain new isolates when we find them. And that is, of course, when we do enough annual virulence surveys, and ideally we do them globally to get more information. To get to certain effectors, we would like to generate a pangenome, and we are in a good position now to look at the North American isolates to develop that, and from that an effectorome, and correlate that with the populations. As I mentioned, we can do that through a genome-wide association study. And also you can look at natural mutations. Some of the avirulence genes were cloned because of differences they saw when sequencing natural mutations. The last one is using a genetic map. I don't know if people are familiar with how this screening is done normally when virulence isolates are collected. They are often in the winter phenotyped on differential cultivars that have single race-specific resistance genes. Here's one example. You often see this 4 or 5-letter code. Here's an example. This B here means on the second place. This means it's screened on this second set. And the B stands for here low, 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 high. And low means it's avirulent, infection type 1 to 2, and high is when there's pustule forms. You get infection type 3 or 4. You get a score of 1. So in this case, here is an isolate from 2002, and later on it was closely related in the phylogenetic tree. We find that in 2005 there is a different scoring. So on this particular set of differentials, you see that they became virulent on another isolate, another differential cultivar. So the letter code changes. And that way we have, when they're all screened, all these isolates, we have virulence profiles. We can correlate that now with the different genetic information we have from the SNPs that were called. And then in the haplotype genomes, we can look for changes in protein presence or absence, or even expression. That's why we also have RNA expression data for some half of the isolates to see if there is different expressions. This could happen, for instance, if a transposable element jumps into a promoter region of a certain effector gene. We also work with a mapping population. This is an old mapping population that was generated by Szymborski and Dick in '68. It was revived by Brand McCallum and Barbara Mullock. And we have been sequencing all the progeny and the F1s, and now we also have haploid-based parental genomes of this race 1 and race 61 parents. And we're currently have a genetic map where we have a number of isolates or a number of markers that show linkage to 3 candidate avirulence genes. So we have to look whether they're in proximity or on specific effectors in the genome. Of course, when we have this potential isolates, we would like to do functional assessment. We did a quick study using foxtail mosaic virus vectors that we got from Kostyuk and Yuka. It works for certain isolates. So we cloned here the previously cloned AVR SR35. This seems to work. We can see here the symptom formation on the leaves when it's expressed through this foxtail mosaic virus system. We see by DAP staining, we see the ROS production and also the PR1 gene, which is indicative for a hypersensitive response, is way up in the combination of the AVR1 gene with the cultivar that has the SR35 gene. However, we tried other cultivars and we also checked the AVR SR50 gene, but that did not work because not all wheat cultivars support the viral replication. So that is unfortunate, but that needs to be tested first if you want to test your effectors. We also have developed another system using Euslago hordeae. That's a barley pathogen, but it's non-pathogen of wheat. It causes very minimal symptoms on wheat. And we created gateway-specific vectors where you can insert your gene, your effector of choice, either driven from the Eustalogomatus heat shock promoter, which is a constitutive promoter, or from a promoter from a effector from Euslago hordeae that we cloned. We can show expression, and when you delete the signal peptide, there is no more secretion. And we can do this in a single haploid strain of Eustalogo hordeae, but also we have what's called a solo pathogenic strain, which we got through a collaboration with Günther Dollemann in Germany and Bilal, which is basically a haploid strain which has opposite mating types transformed into it. So it's a filamentous pathogenic strain, but it's a haploid. And it's easier to work with because it resembles more the pathogenic cell type. So we tested this system with 3 different effectors, the Staganosporum nudorum tox1, tox3, and also the Zymoseptoria triticei effector. And we do see the formation of hypersensitive response necrosis formed. Here you see the fluorescent materials. We checked the PR1 expression here as well. However, we also tested here the ROS production in a luminol leaf disc assay. And unfortunately, it does not work till now for these 2 candidate stem rust effectors we had. And we don't know exactly why not. It doesn't seem to easily produce the protein. So we're working on figuring out why it does not work. Eustalogo hordeae does deliver its effectors in the cytoplasm, which we know from our work on the Eustalogo hordeae AVO1 gene. So it is not quite clear at the moment why this system doesn't work for these 2 rust effectors. So a quick note on fungicides and biocontrol that also is using our genomic information. We published this quite a while ago, but I still would like to mention it because it seems to be falling a bit by the wayside, is suppression of leaf rust disease by expressing hairpins, small interfering RNAs in wheat. I think one of the main problems is that people don't like to make transgenic wheat with these silencing molecules, but it was quite effective. We wanted to follow this up. So this is a single targeting single genes, MAP kinase and a cyclophilin gene, but we also have transgenics in transgenic transgenic wheat plants that have also both genes in their genome. So they express both genes or target both genes in the rust fungus. But we were not able to test them just because lack of funding. But I know that people have tried SIGs as well, so spray-induced gene silencing, but I don't know if that is really viable on big crops like wheat. Another little experiment or project we did was with a company, Terramera, which is now called Catalera, is where we looked at novel formulations, so additives, what they called actigates, based on natural compounds and the derivatives to enhance the action of commercial fungicides. And in this case, we set up a pipeline. This was work done by Mang Li, mainly, where we look at the compounds when you add them and see if they inhibit spore germination. We have a detached leaf assay and then a whole plant assay. This is all set up in a sort of a automated spray and analysis chamber using computer vision. The bottom line is that some of the actigates, they call them, for instance, a compound from goat milk, a natural fatty acid, can reduce the effective fungicide concentration by 75%. So that would be a major saving for producer if that is used. Here we did tests, 2 different commercial compounds, proline and Quadris fungicides. We also looked at the mode of action using transcriptome analysis. And to make a long story short, is we do actually see that some of the additives are actually enhancing host defense responses, even a systemic acquired resistance response. So this is an interesting development that we're following up on. So just to summarize then, what we could do with the surveys is, and I think this is one of the goals of this expert working group, is really to increase genome sequencing of populations or isolates from different places in the world, and to expand the clades information and look at the B allele, the mating type alleles, and also to be able to get a better feel of what the world population looks like. This would need a lot of effort from a lot of pathologists around the world, but I think this can probably be done. There is already quite a lot of information available. It needs to be put together. As I mentioned, if we have this kind of information, we could screen for the homoheterozygous state of avirulence or virulence alleles, genes, if we know what they are, and get potentially reliable and fast functional assays or PCR-based assays. If this is combined with a biovigilant approach using air current modeling and maybe even phone apps, I know that some of these developments are happening in other regions and with other pathogens. So, vector genes based RNA gene selection and cloning, so we can use them in breeding programs by stacking, combining both race-specific and race-non-specific, or even engineering our genes. We could potentially identify susceptibility genes. There's already several examples in the literature, and that was based on bioinformatic analysis of transcriptomes. Of course, the idea being that we could probably modify them by maybe by CRISPR technology, and also manipulating systemic acquired resistance. One of my students is involved in this, and I hope that she will be able to next year maybe give a seminar about this development, which is a very interesting approach. I quickly touched on that we could maybe find bioactive active compounds to be able to use less fungicides, and I quickly brought up again the use potentially of RNAi. And then the last development is interesting over the last few years is groups are looking at how the microbiome on the leaf surface could maybe be studied, or is being studied, can maybe be used to combat rust diseases. So with that, I already mentioned quite a few of the people here that were instrumental in doing a lot of the work. So I really want to point out again David Kim and Sean Formby for their excellent work on the phylogenetic and phylogenomics work. My longtime collaborators for the pathogen collections. John Feller is also longtime collaborators for all the isolates in the US, and some of the other people here. And with that, I thanks for your attention.
Thank you. That's really interesting and elaborated talk on the rust and genome.